All right, we'll get started. Ready? Okay. Okay. All right. So the back here, I try to yell because my sound may not be strong enough. Once again, people in the back, if you cannot hear me, please come to the front row. All right. So last time, my last lecture, we discussed the recurrent network, some property of a recurrent network. We especially focused on the half-wave network, which is a very typical example of a recurrent network that allows you could call point attractors. So basically, discrete states that are stable. Right? So this insight was that those dynamically stable states correspond to the memory of the system. They have lots of good property. Like if you have inputs that's corrupted, it automatically cleans up the noise and recover the stored memory. So in that case, it really looks like a memory, right? So in today's lecture, I'm not going to talk about recurrent network, but it's called continuous attractor network. So basically, the idea is very similar to a half-wave network. The only difference is that your stable state, instead of a discrete patterns, which are good to represent something that's discrete, now you have a continuum of possible stable states. It's almost a continuum stable state. So that's good to store something like spatial memory, like location in a space, which can vary continuously, right? So the story is very interesting. It starts from this patient HM. I'm sure you learned this patient in your previous lecture, right? About a patient who had a surgery in his late 30s, in his 20s. In his 20s. He got both sides of his hippocampi removed. You heard about this guy, HM? Yeah. So it's very unfortunate. So once this happened, so basically he cannot form new memory anymore. And it's what particular type of new memory, right? He can still remember how to swim, how to ride a bicycle, how to tie a shoelace, stuff like that. But he cannot form new memory about the facts and his experiences that he can talk about. So basically, as you remember, the doctor who worked with him for several decades. So every morning you have to introduce himself to this patient again. So I'm Dr. Who and Who. He says, hey, how do you do? You know, he didn't even remember that you've met this doctor before, right? So it's a very serious problem. But this person, if you talk with this guy, he appeared to be perfectly intelligent. He can carry out the conversation just like a normal person. But once you switch the context, you change your topic, he forgot what happened before. Just a moment ago, he just forgot immediately. It's a very bad sort of a situation. Like, basically, this person is trapped to the present, right? He has no future, has no past. Well, maybe the distant past is okay. So the past that before the surgery is still okay. So that's the interesting part. He can remember his things about his high school, but just after the surgery, you no longer can form the long-term memory. All right, in the past, the thing's still okay. So this gives people the concept, right? Your memory, maybe you just store somewhere. It depends on the hippocampus, but hippocampus is more like, if you know your computer, it's more like a RAM, right? Like your computer, you have a random access memory, right? There's also long-term storage. It's a hard drive or solid-state drive. So, especially the old computer, if you plug the power, if the battery is removed, all the temporary information stored in your RAM and before it's stored in the hard drive, will lost permanently, right? But your memory is stored elsewhere. In the human, we think in the neocortex. That's still okay. If they're already stored there, it's still okay, right? So this is the sort of a Hollywood movies, right? There are lots of movies that plays on this kind of deficit, so memory deficit. So this is kind of a very cheesy kind of movie. It's not very good. But this one is actually more interesting, the Memento. So if you haven't watched it, it's worth watching this movie. It's a very low budget, but what's interesting that this director tell the story in a reverse order, right? But locally, it's a normal order. It's like a story. So basically, the final effect is that if you watch the movie, you would feel like a patient, like HM. If you lost your hippocampus, that's what you feel like. For a short time period, everything makes sense. If you're perfectly normal, intelligent, but when your context switch, you suddenly, you forgot everything right before you. It's like a horrible. You don't want to get into this kind of situation. But today, why this guy hippocampus get removed? You know why? Because you have learned in your past lecture, I believe, the hippocampus is the most plastic part of the brain. In the sense that you look at the NMDA channel. You remember NMDA channel is a channel that's important for synaptic learning. Those channel has the highest concentration in the hippocampus. So this part is very plastic. But once you have something look like a habit learning that we talked about before, things fire together, tend to wire together. The hippocampus has a lot of those kind of channels like LTP. So if something happens in the hippocampus, that pattern will be strengthened. Right? The cells that fire together tend to be wired together, right? Because they are wired together, they're more likely to fire together in the future. So they get like a positive reinforcement loop. The things get strengthened more and more. If that's the case, the system become self-sustaining activity all the time. So that's called a seizure, right? So that's why the hippocampus is a location where a seizure most likely to occur, because it's a very plastic part of the brain. But now we know that when you're in a seizure happening in the hippocampus, you don't want to remove the hippocampus altogether. Otherwise, you get this kind of horrible deficit, right? So basically, the people, you change your life altogether. This also gives us an idea, right? So basically, your life is really made of memories. You think that you have a past, you have a present. The present is very short, maybe a fraction of a second, and moving on all the time. You leave this in a short little, maybe less than 1 second. And you can think about the future. The future is not here yet. You can have a plan for the future. But once you have your plan down, the plan is stored in your memory. Right? You live only for that like 1 second or so. So once your memory is destroyed, you think that everything will be different. Do you want to go to see a movie tonight? Do you want to go to a university for education? Why do you want to do that? Afterward, you forget everything about it, right? Is it worth doing? That's really that just really shows you very clearly. It's really most of the stuff in your life is made of memory. So what is this hippocampus? Right? That's because this deficit HM is just a single person that make people so interested in this hippocampus, because this deficit is totally amazing. Like people in the past never realized there are many different kinds of memory. You can only selectively affect this kind, but other kind of memory is still okay. The hippocampus, you look at this Van diagram for this is a macaque monkey, it's similar to us. The mostly visual system on the right-hand side of visual system. This is a highly simplified diagram. It's not a real thing. It's more complex than this. You start from the eyes. This is LGN. So this is the thalamus, right? Information go to the different parts of V1. There, MT. This is the motion area. This is V4, the formal area. So hippocampus is right here. The top box. The one next to it is called entorhinal cortex. That's where all the information from the cortex is all the visual system that go eventually to here, to the hippocampus. So hippocampus is most far away, based on anatomy alone, most far away from the sensory input of the brain. You cannot go any higher. That's it, right? Oh, you may imagine, if this is so high, maybe this is the seat of the soul, the so-called soul. It might be sitting on the top, try to, steer the whole machine moving around. Right? But it's not, right? If you remove the hippocampus, which is cell, you're still intelligent, but just your memory has very horrible trouble, right? You're still intelligent. You can talk, you can have language, everything's okay for a short period of time. So this side, this is somatosensory cortex, right? But these are actually this is a motor system. So basically, this is the output of the brain. These are the input, sensory. Eventually, something happens. Eventually, you go to output the motor response. Right? So the things you don't need to respond immediately. For example, I look at there's a restaurant. It's very good. That means that I have to eat again in this restaurant, right? But you may come back at a later time. So you don't act on the information immediately. That you need the memory. Right? You store that to use at a later time, right? So because of the memory deficit of hippocampus, people start to record from animal, because you cannot do that to human. With human, you get a tragical consequence if you should touch a hippocampus. But they do this in the animal. So the animal, when they have electrode, to record from a single cell in the hippocampus of a rat while the rat is freely behaving, do all his stuff. Right? What they discovered is kind of astonishing. So what they found is this, the first cell they found called a place cell. So the neurons seem to like a particular location in the environment. So this is not a very rare kind of cell. It's in the hippocampus of a rat, a typical cell in the hippocampus is a place cell, right? If the rat is running around in the room. I'm going to show you more example of this. And another two types of cells, it could a head direction cell, which is like a compass direction, like sort of a compass direction of the heading of the animal. And they also like a grid cell. Grid cell is a periodic cell. It's like a tile of the environment. So kind of totally unbelievable. So I think last year, the people who discovered the place cell and the grid cell got the Nobel Prize in physiology and medicine, right? You remember this one? They said, you know, the popular press says they discovered GPS system of the brain. Actually, it's not GPS, but it's something very amazing. How is cell? I think the one thing we have to say that these cells represent location in a word-centered coordinate system. Right? That's very important because all your sensory system is not like that. You must need to do some computation to figure out your location in the word coordinate. I'm going to just give you an example. So why these cell fires? They can recognize location. They're not used based on GPS, like a satellite outside the orbit, but they use two kinds of cues. One is the landmarks. That's for in a familiar environment, use landmark. Another is the path integration. It's based on self-motion. The fact you're moving, you can just count your step, right? It's called dead reckoning when people in the past do the navigation. So they use both kinds of information. It's a very clever system. So first let me show you what the data looks like. This is a typical data. So this is a typical set of experiments. What they do is that they record a neuron. Of course, the animal has this cable, and because now people can do wireless recording, but this is the standard kind of thing. You basically have an electrode, the recording from a single cell in the hippocampus or some region in the brain. And you also have a camera on top, so you know the location of the animal. So you see there are two lights here. Sometimes you use infrared lights, sometimes you use a real light. You use two lights so you know the location and the heading direction. All right. I'll first show you a video of a head direction cell, because this cell is the simplest one, because its representation is only one-dimensional. So what you see is that they're recording from a single cell. The cell fire, you see? The fire. Now it's quiet. The animal is moving around. Nothing happens. It's very quiet. You see, whenever animals head facing in some direction, cells start to fire. In this case, the environment, you see, it depends on something. So this animal is that this environment is a cylinder, right? Because they want to control the cues. What's the environmental thing? They call cues. Basically, all the factor outside what control the firing. So this one you see you can notice on there's a piece of paper on the wall. That's the only landmark in this environment. Otherwise, it'd be completely symmetric. You don't know where you're located. This is the only landmark for the animal. So if you plot the activity of that neuron as a function of animal's head direction, you get a curve look like this. It's like a beautiful Gaussian curve. It looks like a fake, right? But it's real. It's the signal to noise ratio typically is amazing, right? You have a really good firing. So there's a preferred direction of that cell. If the animal's head pointing in that direction, the cell fire at a very high firing rate. If you move away, the firing rate drops. Right? In the background, there's some firing rate, but it's much lower. So one thing that's important, you have to realize that this direction is really direction in the word coordinate system. So if the animal, it's not really the animal's head, where the animal's head relative to the shoulder. If that's the case, it's very easy to build a sensor. Only sense what's your angle of your head relative to your shoulder. It's just some stretch sensor or some sensor. But this is different. So if the animal moves in an environment like this, if that cell like the north direction, then it will fire in these three locations. It's really the location in the real environment. If you bring an animal to a new room, The animal never exposed, it's completely novel to the animal, the cells still fire in the same direction. So the preferred direction all run in parallel, regardless of animal's location. That's pretty amazing, right? That's in the brain, right? So, of course, you have many different cells. The different cells like a different preferred direction. So these are different cells recorded simultaneously. They not all like the same direction. They like all different directions. So the cells cover all possible directions, right? So the question is, why does the cell behave in this way? Well, your first maybe guess would be maybe there's some kind of geomagnetic cues, maybe the magnetic sensor or something. I know there are magnetic sensors in animals, but this is not based on geomagnetic sensor. Just the experiment can show this. That's what's important is the landmarks, familiar landmarks, also self-motion, because this cell also works in the dark. Let me show you one example, show why this is the case. So suppose this is the cylinder environment you saw before. Let's just imagine this cell like the north direction for this particular cell. All right. So if you remove the animal out of this box and place it back in again, the cell still like the same direction. Right? In a stable environment, the cell have very stable representation, right? So now if you remove the animal, you play a trick, remove the animal first, and then you rotate that piece of card to a new location. That's a single landmark. Then you put the animal back. Then you realize the preferred direction of that cell rotates with the same angle. So basically there's a fixed relationship between the firing direction and that landmark. Okay? So this already ruled out the geomagnetic cue possible explanation of magnetic explanation. It's not possible. It's really based on the familiar landmark, right? So in that case, this may be just a very sophisticated visual system. Somehow I can compute, how that car looks like, based on my location. I just calculate what should be the direction of north, for example. So that's not the case either. So if you start from here again, the standard configuration, what happens if you remove the card slowly? Don't play trick. The animal, while the animal is moving around, you just in front of the animal, you move slowly. The animal realize, okay, you just remove that card. If this is a visual system, you would predict that the cell will be shut down, right? Because that single landmark is gone, right? But the cell actually fires still normally, right? If you turn off the light in total darkness, you track animal by infrared, the cell still fires if in the normal situation. So that seems like very, very strange, right? On the one hand, you show that this card has such a dominating effect. You play somewhere, the thing will follow you completely. It's determined completely by that visual cue, the landmark. On the other hand, you get rid of it altogether, right? The thing still works, at least for a short while, but for like maybe half an hour. If you wait too long in the dark, eventually the thing may actually drift away. So that gives you the hint. So what happens here is that the animal, the firing of the cell is self-sustained. It's not driven by any particular sensory cue. It's firing by itself, right? So what happens is that you anchor the firing with some familiar landmark based on some learning, all right? And if that landmark goes away, the cell fire anyway. So you update the internal representation based on self-motion all the time. So that's a default mode. And then you check out the landmark to try to do a calibration to correct for cumulative error. So that's how people think about how the system works right now, right? So you see that the system, based on what you have learned so far, you learn about sensory system, you learn about motor system. What kind of system is this? It's not entirely sensory, it's not entirely motor, right? It's really, it can be defined by visual cue. Like show here, vision can define my spatial orientation. But I don't rely on anything in particular. So what's happening is that the brain discovered something more abstract. This is a space, more like a Newton space, right? It's abstract space, right? It can be defined by vision, but if you're blind, you still have a concept of space, right? You don't rely on that. You can, based on step counting, there are many different ways to calculate space. So this is one example, the head direction cell. The other cell is kind of similar. This is the hippocampal play cell. The play cell is two-dimensional. So if you record from a cell, the cell will like particular location. This is like a 20 centimeter, about the size of the animal. Right? Some are very big, some are small. So they basically only like some particular location in the environment. So in this case, okay, this is a cylinder. You're looking from above. Cells like this location. This is the, this map shows the firing rate of the animal at the function of animal's location. That's a piece of card. If you rotate to a new location, you see this place field also rotates with the same angle. So this is called place field, basically the firing rate of the cell at the function of animal's location. So then you move the card to a new location, you shift it back, the place field comes back, right? That's very similar. It's also it's anchored relative to familiar landmark, right? So here the video shows a place cell. It's on a track. You see, this is a figure equal figure 8 maze. This is a single cell activity. So this is basically it's just a wooden track elevated from the ground. There's no walls. Animal can see everything, right? Just running on a track. You track the animal's position. This is a computer, the data. The time is not exactly right, but you can see this cell like this location. So these are cumulative diagrams for like 15 minutes. So you can see most of the cell that fire somewhere else as well, but mostly fire around this corner. So basically this cell like this corner. You see the fire other place as well. But here it's very reliable. It's very reliable. So these are the cells in the hippocampus. It's the highest part of the brain. You see the cell, in most other places, the cell doesn't fire much. It's very quiet, right? They're very reliably active only in some part of the location. So here the different cell record simultaneously. Let's just show you that there's not just this corner is special. Maybe you imagine, maybe this corner smells funny. Whenever it goes here, it's just like olfactory cell, it drives that neuron to be activated because it's a funny smell. But this shows another cell. This cell like this location. This cell doesn't fire at all this corner, right? And not just that corner is special. It's every location is special. So there are some cells like all different directions, all different locations in the environment. So this cell like this place, and it doesn't fire much in other locations. So these are basically a typical cell in the hippocampus. That's the highest, very most far away places from a sensory system. It behaves like this, all right? So these are sort of a 25 cell recorded simultaneously in this particular animal. You see we saw this first cell is this one. This one, there's cell like this location, right? There's some cell may like this location. You see other cell may like the middle part, some may like this part, and so on. Some cell may like more than one locations, right? So now you can imagine. So based on the activity of these cells, even if you don't look at the animal, you can figure out where the animal is located, right? Suppose this cell is activated, this one, the animal must be somewhere here around this corner, right? If this cell is activated, you mean the animal may be somewhere here. So it's basically, this is sort of a representation of different, the activity of a cell, represent all different locations in the environment, all right? So any questions so far? Anything unclear? All right. So here shows the head direction cell and the place cells are tightly coupled, in the sense that in this particular environment, you have the one cell like this location, the other cell maybe like this location or some other places. There's head direction cell. So here it shows a angular, this is called the polar coordinate system, right? So the firing rate of that head direction cell is a function of animal's heading direction. So basically, the head direction cell, this row is head direction cell. Okay, they like this direction. There are two place cells, here and here. Now you have a different trial. You record it again at a different time. You get the cells kind of reliable. It's basically very similar. Place cell, similar location. The head direction pointing in the same direction. So in this case, what happens is that they spin the animal around, try to confuse the animal, right? To somehow make the animal very dizzy, so you didn't know what's going on. Then you put it back. In that case, you see the place cell used to be here, that rotate to a new location. Right? So this place cell also rotate about the same angle. So you see the head direction out here is also rigidly rotate the same angle. So basically, what it shows you is that these place cell, high direction cell, all the cells, they form a very rigid coordinate system, right? They can be wrong. In this case, sort of it's wrong. So basically, your internal sense of direction and the external cue in conflict, right? We all have been disoriented sometime in your life, right? Remember, sometime you got disoriented. You try to find the house in a new location. Eventually, it's maybe wherever you think is the north may not be consistent with the map, right? Sometimes your sense of direction can be very different. But you never get this. You never be confused. Whatever direction you're saying, this is the north. I already know the opposite is the south, right? You're never confused about that. So you have an internal rigid system. The system may be wrong relative to the outside world, but your internal system is still very tightly coupled. Right? That's shown just here. In this case, the animal is wrong, but the internal system, if it's wrong, everybody wrong together. Right? That just shows this tight coupling. So the third type of cells that we discovered more recently is this gray cell. The gray cell is truly very baffling. It looks like unbelievable, right? This is the record this activity of a single cell. These red dots as the spikes, right? The black trajectory is the animal trajectory. So this cell fires in different locations, right? This is the sort of average farm you see. The weird animal active form a hexagonal or triangular pattern on the floor. Is that amazing? If you haven't seen this thing before. Remember, this is from a brain of a freely behaving rat animals moving freely, right? Completely free. You're recruiting from a single cell. Here, look at 3 cells simultaneously. Right? These 3 cells, they all are hexagonal, but if you plot them together with different color for the 3 cells, you see the red one, they form a hexagonal pattern, the blue one, another one, the green one. So they all have, the cells in the neighborhood, they all have the same spacing, same orientation, right? But the spatial location, because spatial phase may be different. So all these cells, they have like a tile, they tile the entire plane, right? So this is completely amazing because you think about space. You think of space is continuous, right? You can move continuously, but whoever predicted that inside your brain, the representation of space, this is entorhinal cortex. And you remember from the anatomy, that's the location next to the hippocampus. That's the lowest one. Yeah, this is 4 different kind of gray cells. Some cells, the spacing is small, so this is a 50 centimeters. Some are large. So there are many different sizes of the grids in the brain. You may think this is just maybe just rats as kind of a weird animal. You have this kind of a geometric pattern in the activity in the brain. Maybe we humans are much smarter. We're not like this. But it turns out a more recent study of humans do virtual navigation. It's like play a video game, like a taxi driver. You pick up people in the street, and then you can plot activity in your brain in some parts of this entorhinal cortex, and it sees how that correlates animal with your directional movement in your virtual environment. It turns out you see the 120-degree periodicity of activity in the entorhinal in the functional imaging study. So it's most likely human have this kind of system as well in your brain. Right? So that's kind of very weird. It's like when you first see stuff like this, we just say, well, of course we understand, we don't understand the brain. This shows you very clearly, right, how little we know about the brain. How could be something so regular? It's almost like a mathematical thing for something, they're computing something. It's like a basis function of something, right? So like I'm not sure it's a Fourier series, but it's all the place, all the other stuff constructed based on something look like this in the brain. It's very strange. So anything unclear about this? Can people hear me in the back? I'm not sure. It's okay? All right. So how is this possible? So I'm going to show you some theoretical ideas. Right? There are several theoretical ideas, but this theory I think would be probably the leading theory now. The people most typically think about this way. It's called this attractor model. So all these 3 high direction cell, place cell, gray cell, they are kind of similar in the sense that they are word coordinate system. If it rotates, a landmark, everybody rotates. They're all tightly coupled, right? In a normal situation. So the idea is this: if I use the simplest one, the ring attractor, because it's one-dimensional for the heading direction, right? Let's give you an explanation. What's the key idea? So you first look at the data. Let's just put the neurons. So each of the cells is a head directing cell. They like different direction, right? If we just arrange them according to their preferred direction. So these are the cells, the preferred animal facing north, all right? So these are the cells facing this northeast, right? And so on. Right? Just plot the data this way. In that case, you can imagine when animal is facing a direction, you have a bunch of cells. You can plot it as a single line. There's be a bump of activity centered somewhere around animal the current direction. When animal head moves, this bump activity moves with animal, right? So basically, this is just a way to visualize the data. So you basically have an internal simulator. You have a ring of cells in your brain. Whenever your physical head is moving, there's internal representation moving with you. Try to simulate what's going on, right? So this can be done based on because this activity must be self-sustaining in the sense that if I turn off the light or remove any particular sensory cue, the cell still fires, right? So that means the activity must be self-sustaining. We can get self-sustaining activity in like say half-wave network, like a recurrent network by recurrent connection. So the idea is that the cell is active. Why is active? Because their friends excite each other, the whole thing, hold themselves together. So in this case, it will look like this. So you have a bunch of cells, arrange them according to your preferred direction. I assume that the cells, they excite their neighbor nearby. You excite the cell close to you, but you may inhibit stuff far away, right? But of course, through some inhibitory neuron. So you can write the equation look like this. So your activity now depends on two variables. One index is the location x and time, right? So in the past, when we talk about this, location x is basically which cell. It's basically the index of the cell, right? But it can be continuous, right? If you have many cells, you can use it as a continuum. This equation is almost similar to the continuous version of half-wave network, except for here, there are the weights. So we assume that the weights here depends only on the relative location, but not absolute location. So that means the space is the same everywhere, right? So how the two cells should be connected only depends on relative position. It doesn't care where it's actually located. Right? So then you can write the thing like this. This is exactly the same, with the assumption we call the shift invariant, right? So what it shows that, so this connection is sort of symmetric. So the cell, you treat your left neighbor and the right neighbor in the same way. So this turns out to be the same as last time I said the weight matrix of half-wave network is always symmetric. I mean, if I ever connect with you, you connect with me with the same weights, right? So this is the same thing. If your left and right is the same, and it's a shift invariant, right? If this x goes here as there, the backward connection must be the same, right? So this is the symmetry is the same as reciprocal connection is equal, right? So if you have a system like this, how does the system behave? It behaves like this. So you start, this is the line is all the cells. You start with some random initial condition, right? The cell will evolve in time. So you may see the system is sort of symmetrical. Everybody connects with everybody in the same way, except you excite your near neighbor, you inhibit your far neighbor, but the situation is the same everywhere, all right? But you may imagine maybe everybody, the activity, everybody equal, like a flat activity state would be the equilibrium state. It is equilibrium state, but that state is not stable, right? It turns out stable states is a symmetry breaking. It is a peaked activity like this, right? It can be centered in different locations because your situation is the same everywhere. If I can balance this bump activity here, you can balance anywhere along that ring, all right? But here I just draw as a line. The 0 point and the 360 degrees is the same point, all right? All right, so this you can explain why the activity of this neuron would be self-sustaining because this is because attractor, the cell, their activity here because this bunch of cells activated, right? And another thing is that you see you have a two bump simultaneous activity together, but this is also not stable. One will win the competition, right? If there are any slight difference, will win the competition. If you have a very flat state, if just one cell slightly different from other cells, that cell will be very active. Everybody will push down. It's just like in the society even if everybody had equal right, just based on the trading, random initial condition, someone will get very rich, and other people will be pushed down. Even you have, that's also possible, right? Even if your rule is very fair, eventually the result may not be fair, right? Like this. It's only, everybody equal, that's unstable. Right? You get any perturbation that anyone will get more money, it will be, will accelerate. That's get even more. So this sort of behaves in that way. So the system, if I show you this sort of the absolute function, the energy function, sort of interpretation would be, say the last time the Hopfield network would be you have a sort of a discrete state in your possible activity that is stable. But here that you have a continuum of state. This would look like a bottle of the bottom of a wine bottle, right? You have a continuum of possible states. But these states, each one of them would look like a stereotype, a Gaussian kind of activity bump, right? That's sort of how activity would distribute among the neuron. But you can rotate the activities continuously along all these rings. So basically, along this ring state, they have equal energy. Everybody equally sort of stable. It's more like you put a ball on a perfectly flat table, right? You can put it anywhere you want. But it cannot be, so other state, as I said, would be, it's not that bump. It would be 2 bump or 3 bump or other sort of random shape. Those are not stable. They're going to settle into that bump somewhere around this ring, right? So this is what we use to the dynamic system to explain this phenomenon. But how can you move things around? So I just give you very quick ideas that if you have this connection, what happens if you say, I shift this activity, shift this connectivity pattern. So this curve shows that given this cell in the middle, how it projects to its neighbors. It's no longer symmetric. I excite more my right neighbor more than the left neighbor. If you do something like this, the system will have a traveling wave of activity. It's like a soliton, sort of like, except for there's no collision. But you have an activity, even you start from a random initial condition, the activity will emerge, right? And the traveling at a certain speed. So that speed of traveling is actually proportional to how much you shift that weight. Right? So that also works in 2 dimensions. Is that clear so far? Yeah. So this also works in 2 dimensions. 2-dimensional case, let's imagine you have a bunch of cells you put on a square, on the square grid, right? So the height means activity. You wire things up in a similar way. The cells close by excite each other, right? They inhibit things that are far away. So in that case, you start from initial random condition, you will see that there's a bump will emerge. If you shift your weight, not symmetrical, you excite your neighbor towards one direction more, this activity bump will move, right? So the whole mental picture is like this. If you have an animal, let's say here, running around in a room, or this is a rat, right? Let's imagine you can put all your place cells in the hippocampus on the floor, right? According to their preferred location, where the place fields appear. If you do that way, you can imagine when the animal is here, the cells, they like this location will be activated, just by definition, and the way I arrange the data, right? When the animal moves, that activity bump will move with the animal, right? So this would be similar to the, you use the ring to represent all possible direction. But here you have a simulator, like a 2-dimensional thing in your brain, in the hippocampus, it represents animal's current location. When animal moves, you simulate what happens when the animal moves. Right? So here's the interesting thing. You think that this case, what happens when animal moves outside of the room, right? If a place cell, you may generate more representation here, but there's one interesting idea that if you can, maybe you can connect. In your studying mathematics, you have sort of a periodic boundary condition when you solve differential equation, right? If you connect these cells along this line with that line, you will form a sort of torus, right? So if you sort of this is a sheet of cells. I suppose you connect this part, you make a sort of a cylinder, and then you connect the cylinder, right? The whole thing becomes a torus. So in that case, if I record from particular cell here, when the animal moves forward, when the activity moves to here, then the activity will reappear over the other edge because that's connected, right? When animal moves in the real world forward, but inside the brain, the activity will move along that torus, right? So you have periodic firing just like a grid cell. We have a periodic, but in this case it's a rectangular grid. You get this grid cell. It's a little bit harder to explain, but that's the idea. reasonably clear? Or anything unclear? All right. So the comparison of different animals. So so these are different animals. We know that in rats, in mouse, and in bats. The bats is interesting because the bats is like look like a rat, but it's really flying. They're living in three-dimensional space. So what in their hippocampus, they find that they have a place cell in 3 dimensions. In the monkey and the humans, so we have a place cell, but in monkeys, it's more like a view cell in the sense that you think about space. For a rat, if I want to explore a location in space, I have to move my nose to there. I have to walk to there, right? For like us, like monkeys, we're a visual animal. Like most information comes from visual system. The rats, they are more olfactory, more important to them. Or maybe they're like a tactile, their whisker is more important. For humans or monkeys, we can look at this thing. It turns out the cell in hippocampus in the monkey seems like it depends on where the animal look. Right? It's not really driven by sensory cue. You turn off the light, if you look in that direction, the cell still fires. So it's really my terminal location of my gaze. So that's more complicated, but there are some difference of these, but there are some similarity as well. Because they all work center nervous system. If you turn off the light in the dark, the thing still works. It's a very amazing thing. So this is that flying bat in a room. Let me see. This is so the bat uses you can use this like a very big bat. It's the Egyptian bat because the bats are big here because they can carry a lot of weight because they use like a wireless recording of animals flying. So they have some artificial trees here. Animals flying around. You see this case. This is 3-dimensional in that room. The cell like this location. Right? So the cell like this location and so on. So this like really have a 3-dimensional representation of space. So the cell in the hippocampus in the flying bat, they like some particular location in a 3-dimensional room. Right? It's really this works in 3D as well. Right? And more recently, I didn't show you data that people in the Janina farm, they showed that in an insect, in the Drosophilus, the fruit fly, in their brain, they have cells. They actually have a ring of cells that represent different heading direction. So in that case, they are. You can actually see that activity ring actually directly inside the brain. This is the human hippocampus. So that's the Jennifer Aniston cell I sort of mentioned to you. So in humans, people, we never try to make the people sort of move around too much because the people, they usually, they show them different pictures. So one finding that seems very robust is that the cells in the high-level visual system and the hippocampus, they like some individuals. So these are the example of a Jennifer Aniston cell. So this is, but I said this result get a lot of popular press, right? They show this kind of, because this is sort of people understand this very easily. So that's her photo. So this activity, the raster diagram of the cell in different trials. This is sort of histogram of the activities. This is a cell like her. So here that, yeah, this is like a Brad Pitt. So the cell really inhibit a little bit. There are some other actresses here, right? So the cell likes her. So this is a very strange thing, right? Because you think about this. So you look at the picture. So each one of the picture look very different. Right? They're very different. If you look at this picture, say the dark background and some like a face in the middle, it's kind of similar to this one, but the cell doesn't care. But it's a different individual. Right? It's really care about that person, even though the particular the visual look is very different. Right? So this is sort of like a place cell in the sense I really like that location, but that location is defined different. If I remove the landmark, the visual look is very different, right? But that's still that location, right? But this sort of among all possible space in the space of possible faces, like that particular spot, which is corresponding to Jennifer Aniston, right? But this shows you two things. First of all, the cell is very specific, right? They care about her, but not other actors in a similar, similar dress, similar look. It had to be her. It's very specific. And also, so this phenomenon, this has to be learned, right? There's no way you can born with a Jennifer Aniston cell because this is it has to be learned. So in the past, there are always ideas. How to represent something specific? There are two different ideas. One is there was a name called grandmother cell. It's meant to be a joke, right? If some person, you only have some cell that only represents your grandmother, but nobody else. The other idea is that you use like a population representation. I'm going to cover that in my future lecture. But in this case, this is really like a grandmother cell, only for a particular person. So there's more example I show you. This is called a Halle Berry cell. That's another actress. Right? So that's her. You see that the cell responds to her, even her picture. Right? So not even like here. This is like the words said Halle Berry. This is like an English word. So it respond a lot, right? This is there's no mistake. That's her. Wait, some of it. It's really specific, but they're all very noisy too. There are some other cells. They like some location. So this is sort of a Sydney Opera House. There are also other cells. They like Pisa Tower, the Leaning Tower, Pisa, and so on. So they also cells. They like a particular landmarks. Right? So all these things shows that. For the hippocampal system, even though we have some model, is that we know how to do things like this, right? The Sydney Opera House versus the words, we can do association. Right? If you learn the thing all together, I can activate any part of it. You can activate the whole representation, right? That part we know how potentially you can use a sort of a recurrent network to do this kind of stuff, and also But exactly I'm sorry I cannot give you a coherent picture of how this whole thing actually works. We have very interesting information about some part of it, but we don't have the whole story yet. But from what we learn, we know that if I ask you to represent some discrete fact like this, you can easily use discrete attractor to do that. Or you can represent something continuous. We think possibly you can use a recurrent network to do that too. The idea is actually very similar. So any question? Anything unclear about this? Yeah, just here first.
Do we have any idea of how it's assigned for that specific thing? So for one person, how important is Jennifer Aniston in her life? How would I know what people have neurons in my brain?
Yeah. So that means how do you know? Yeah, I think first it must be learned, right? So the people, when they do the experiment, they interview the people. What's your favorite actress? Whether you already make sure you cover the stuff they care about. Otherwise, I show you a bunch of pictures from other countries. You have never heard of the people. There's no way you represent them, right? So the people that your friend, your family member, I'm sure you have a cell devoted to them. That's almost guaranteed, right? Every single thing you can recognize, there must be some cell responsible for that. So another question there?
These are recordings of individual cells, right?
Yeah.
How do you find which cell to record from?
Yeah. So these are these are recorded from individual cells. Right now, there are no method to do that. So basically, they just record many cells at the same time. So the current state are about 200 cells at the same time, just randomly, just by chance, So there is a brain initiative. You heard of brain initiative? They're pushing to record maybe 1 million cells at the same time. I'm not sure it can be done in 10 years, but so that's the thing. You can see that neuron, if it's devoted to something so specific, you really need to record many neurons at the same time, right? Otherwise, you will miss a lot. Yeah. Any other questions? Okay, that's all. Thank you.